So here's our Phycomyces plate that was inoculated 1 week ago. We have our plus strain over here, and we have our minus strain over here. And you can see right down the middle all those little teeny tiny black dots. Okay, so basically our plus strain grew out, our minus strain grew out, and that's where our sexual spores are. We're going to examine that line of dots when we go. Okay, now we have our plate underneath the dissecting scope. You can kind of see I have it focused in on some hyphae way above those little black dots. Those little black dots then are what we're running down the middle of the plate when we looked before. And so those are going to be the sexual spores, and those are what we want to focus in on. So a big question to start off with: what is the purpose of sex? So why do organisms have sex? Okay, now I ask this of students a lot of the time, and a great deal of the time I get the answer that sex is for producing new individuals. Now if you wanted to produce a new individual, it would be easy just to kind of grab some of your cells, throw it on the ground, and have them grow into a new individual. Okay, evolutionarily that is something that could easily happen. But in reality, the purpose of sex and sexual reproduction isn't really just reproducing. It's basically getting individuals with genetic diversity. So genetic diversity is the goal. And for this week in the fungi lab and next week in the plant lab, I want you to really think about how these life cycles are adapted so that we get genetically diverse individuals. Now, genetic diversity comes through meiosis, right? So meiosis is a critical part where basically we take, chromosomes from one parent and chromosomes from another parent, mix and match those chromosomes so that the baby has different chromosomes throughout them. We also have something called crossing over where, right, the ends of the chromosomes swap, so each chromosome then is also genetically diverse. Now, a couple other ways of getting genetic diversity beyond meiosis, right, of course, is when you're thinking about breeding. So basically, having sex with somebody other than your relatives, right? So fungi are exactly like people in that fungi also don't want to have sex or don't want to mate with their brothers and their sisters and their relatives. So to avoid mating with their brothers and sisters and relatives, then it's important for them to disperse and get far away from their original location so that their probability of mating with a non-relative is a lot higher. So a lot of these adaptations for these sexual spores then are designed so that they can do exactly that. So we want to look at dispersal and outbreeding when we're looking at these organisms, and in each life cycle it's nice to look at that. So let me zoom in here, and we're going to look at these sexually reproductive structures. Okay, so this is a zygomycota. We have our plus strain coming in from one side, our minus strain coming in from the other side. When they get together, you can see that there's a big spiky ball right in the middle. Okay, that spiky ball is fairly costly, right? If you're thinking about our non-sexual stages, right, our non-sexual spores occur in sporangia. N-G-I-U-M means sac. Sporangium then is a sac of spores. In this case, the big spiky ball in the middle is called a zygosporangium. So it's a sac of spores in the zygomycota. Zygosporangium. Okay. And the way that the zygosporangium comes about, we can look over here. I'll focus into the lower right. So that's an early stage. We can see our plus and minus hyphae getting together. Then they share their nuclei. And inside them we get this big old ball growing, being our zygosporangium, right? When the nuclei are shared, then the structure is called dikaryotic. So this big old spiky ball is a dikaryotic structure. Inside that, then we're going to have karyogamy occur where the nuclei get together. And then we'll have meiosis occur, creating unique haploid spores inside that ball. Now for dispersal, those spores are going to want to get away, right? And in this case, I want you to think about this. So this ball, as you recall, the plate, is really small. It's down there on the ground. How is this ball going to get away and disperse so that the spores can mate with other individuals? Now, spores are tiny. They can definitely get blown by the wind. But these spores aren't adapted to be blown by the wind, right? These structures are really tiny, sitting all the way down on the ground. So what causes these structures, or how are these guys going to disperse? Okay, why does it have those spiky balls? Now, for some reason, right, we're looking at an investment of energy. And a good policy that we looked at the last couple of weeks is that organisms are designed to save all the energy that they can. So a general rule of thumb is that organisms try to invest as little energy as possible, and then everything that's left over they put into reproduction. But here's a spiky ball. What's going on with that spiky ball, and how does that spiky ball allow it to disperse? Okay, now a lot of dispersal is animal mediated, and these spiky balls are likely to be animal mediated dispersal. So now that we think about animals, right, it looks like those spikes are designed to stick to some type of animal. Now, what type of animal do you think it's going to stick to? Okay, could stick to fur of mammals, but these are really small, flat, way down on the bottom of the ground, so they're not likely to stick on mammals, right? Okay, so, they're really small. And if you look at it, it kind of looks like Velcro. And so the Velcro, you can see if an insect stuck its leg in there, it would easily stick to that insect leg. Now the advantage of sticking to an insect leg is that insects not only have wings and can fly and get a lot further away, but they can stick you exactly where you want to go. So if you go through air dispersal, randomly, you might be able to land on what you want to land on, but you're going to have to make a lot of spores to get those spores to land in the location that you want them to land on. However, if you entice a insect to come in, that insect gets you get stuck on that insect and it flies away. That insect has sensory mechanisms, and those sensory mechanisms allow that insect to find another really good place that's likely to have sporangia there as well. So let's say that this was on a dung pile. You have a nice moist habitat with a lot of nutrients on that dung pile. Insect grabs you, it's going to use its senses and go to another dung pile and stick you right on that other dung pile, so that you're going to be right next to potentially other zygosporangia that have come from other insects to that same spot. And thus these are very specifically designed for getting at genetic diversity and dispersing out and landing exactly where they want to land.